Hello and welcome, dear students. How are you all? I welcome you to the class of biology. Dear students, please report your attendance and we will begin right away. Okay. Centers, please report your attendance. Centers, please report your attendance. Okay, so we are beginning. All right, so yesterday we had done some part regarding the mineral nutrition. So I hope you have, okay. So you have done the nitrogen fixation part. Sorry, we have to begin the nitrogen fixation part. Okay, so mineral nutrition. This was the part we were covering. Okay. So I hope everybody has gone through that part of notes which I have given you yesterday. Okay. So dear students, we will be beginning this right away. Okay. Centers, please report your attendance. That is the last thing I need from you. Okay. So please do this quickly. Centers, please report your attendance. Okay, all right. Itanagar, all right. Okay, thank you, Shillong Senapati, coming along. Thank you for that. All right. So let me start for whatever things we had left for today now. Okay, so let me begin by saying that when we talk about the mineral nutrition part, it is clear to everybody that talking about mineral nutrition takes us to the part where we talk about the deficiency symptoms. We talk about the most of the symptoms associated with the, just a second, we talk about the symptoms associated with the particular deficiency, like zinc deficiency or causing the potassium deficiency, magnesium deficiency. So all this part we have done yesterday. Today the important part is covering the nitrogen metabolism. Okay. So dear students, we are today starting with nitrogen metabolism in plants. Nitrogen metabolism in plants. Nitrogen metabolism in plants. All right. So already we have covered in the last class yesterday that nitrogen metabolism refers to, what does nitrogen metabolism refer to? It is a specific set of characters that are associated with some, just a second, it is a specific set of characters associated with some nitrogen fixing plants, especially who have this capability, right? So nitrogen metabolism refers to those set of conditions that are there. Okay, so let me just, okay, there is a bit of an issue, something is, I want to show you some images. I wanted to show you some images, they are not opening very well. Okay, let me see if this opens. Okay, no problem. Let me give you the direct interface. Okay, so please see this image that is appearing. Can you recognize this kind of image from anywhere? Dear students, can you identify this root which has this pink colored nodule type structure? Yeah, these are the root nodules and it is associated with a particular family. So do you remember these root nodules and which family do they belong to. There is one particular family, and all the members, nearly all the members of this family possess pink colored root nodules. The pink color is due to leg hemoglobin. I'm sure everybody knows it. So what is the name of this family of plants? This is a cross section basically. Yeah. So can you identify this cross section of this family? Or just identify the nodules? I hope the chart is working. This is just a microbial culture growing these bacteria on a agar-agar culture plate. The name of the bacteria is Rhizobium. The name of the family that is associated with these nodules is called a Leguminosae. Okay? Leguminosae is the family which is having this property of forming nodule-like structures on the roots with the help of bacteria called Rhizobium. Alright? So this was what I was trying to show you. No worries. Alright, so let's get back to the point where we have to, okay, so let me start with where I left yesterday. Okay, we were covering nitrogen metabolism, and today it is the part where we have to discuss some things associated with the nitrogen metabolism. Okay, so please tell me, what is the source of nitrogen in the atmosphere? What is the source of nitrogen that is existing in the atmosphere? That is N2, gaseous nitrogen. So please write, nitrogen exists in the atmosphere as N triple bond N, nitrogen gas, dinitrogen gas. Okay. Now this is unreactive. Plants cannot utilize this nitrogen in gaseous form. So they have to make some arrangements. They have to take some changes. They have to take some steps so that this gaseous nitrogen becomes available to them in some other form which can be assimilated, which can be utilized. Alright. So what is that form? What is that form? That form is ammonia. First of all, nitrogen must be converted into ammonia, and then it must be converted into absorbable nitrate compound or nitrite compound. Okay, and then it can be absorbed by the root hair of the plant. This is clear to everybody? So we have to study that mechanism here by which this gaseous nitrogen will get converted into NO3 minus or NO2 minus or NH3. Actually, we will just study the conversion of N2 into NH3, and rest of the part is easy. So converting this N2 into NH3 is the most typical part, and that comes under biological nitrogen fixation. Okay, so you have to write here, please write, nitrogen cannot be utilized in the gaseous form by the plants. Nitrogen cannot be utilized in gaseous form by the plants. Therefore, it is converted by a process called biological nitrogen fixation. biological nitrogen fixation. Okay. And what is this biological nitrogen fixation? Biological nitrogen fixation consists of this chemical reaction. N2 is converted into NH3. Okay. The enzyme responsible for all this is called nitrogenase enzyme. Nitrogenase enzyme. And just now in yesterday's class we have discussed that there is a chemical an ion which is essential as a cofactor of the nitrogenase enzyme. Can everybody remember that? I told you yesterday that there is a metal ion which works as an essential cofactor of this enzyme, of nitrogenase enzyme. What is the name of that ion? This is also a NEET question. Name the cofactor of enzyme nitrogenase. I am giving you options: Mn plus 2, molybdenum, O2 plus 2. Third option is magnesium plus 2. Fourth option is NO3 minus. What is the correct answer? It should be easy one, right? What is the correct answer? Yes, students, what is the correct answer? Please give me the correct answer. Number 4. No, that is not correct. Please see your notes. Please see your notes. Just see the PPT at least. You can just see the PPT also. You can see the PPT also. It's what is the name of that cofactor? Do you remember? Nobody wants to answer. Okay, I'll leave this to you. I'll leave this to you. I'll leave this to you. Okay, we'll solve this definitely. Right now, you must study how nitrogen metabolism is actually going to happen. Yeah, exactly. I'm happy now that you answered correctly. That's the answer. Molybdenum. Actually, answer should have been molybdenum, but plants absorb molybdenum as MoO2 plus 2. So if molybdenum is not given, you can give MoO2 plus 2. Otherwise, either this option should be given or it should be given as MO plus 2 or just molybdenum. This can also be given. Okay. So the answers of this question can be in 3 forms. Either he will write just molybdenum, either he will write MoO2 plus 2, or he will write MO plus 2, or just simple the chemical symbol of molybdenum MO. So these are the possible answers for this question. Nitrogenase must have molybdenum or it will not function. Okay. So I hope you can understand all this. Okay. So let us move on to the next part. All right. One more thing. What are the nitrogen the organisms which fix nitrogen called? Next question. Or just I'll write this as a point. The organisms which fix nitrogen. The organisms which fix nitrogen are called nitrogen fixing organisms. are called nitrogen fixing organisms, and one question will definitely come every year from this part. Now I will tell you all the possible types of nitrogen fixation that you need to know at least, okay, are called nitrogen fixing organisms. Nitrogen fixing organisms. All right. Now let us see about what are the symbiotic nitrogen fixers and non-symbiotic nitrogen fixers. There are some questions are asked from both sections. Name some symbiotic nitrogen fixers. Name some non-symbiotic nitrogen fixers. And the third question which comes, which is the most common question, is name some non-leguminous plants and their nitrogen fixing organisms. 3 questions. Name some nitrogen fixing organisms. Name some symbiotic nitrogen fixing organisms. Name some non-symbiotic nitrogen fixing organisms. Third question is name some non-leguminous nitrogen fixing plants and their organisms. You understand? The most common nitrogen fixing organisms are legumes and rhizobium. Now there are some plants like ulnus, non-legume, still it is able to fix the nitrogen. So what I was trying to show you, that please put the heading symbiotic nitrogen fixation. Symbiotic nitrogen fixation. Symbiotic nitrogen fixation. Okay. So in this you will write the most common nitrogen fixing organisms are found associated with the leguminous plants. You will say the most common nitrogen fixing organisms are found associated with leguminous plants are rhizobium bacteria, associated with leguminous plants. Okay. So the rhizobium bacteria, they are basically associated with leguminous plants. And what are the examples of leguminous plants? Example of legumes. First of all, let's write the example of legumes. All the pulses. Can you name me some pulses? Some dal that you eat? Can you name me some pulses? Can you name me some pulses, dear students? Nobody wants to name some pulses. Nobody eats dal. Nobody eats dal. Okay. All right. So arhar, moong, urd, Phaseolus mungo, Phaseolus aureus, cowpea, chickpea, gram. At least you people must be eating chole. Okay. Chole is just a larger gram variety. Okay. So normal the gram that we eat that is Cicer eritinum. Okay. The normal gram that we eat, the Kabuli chana or the horse gram. Okay. And the chole chana, that is basically a larger variety. And urd dal, moong dal. Okay. All these dals, okay, they are basically legumes. And mutter, everybody at least eats pulao, and I hope that you are well familiar with at least the pea, right? So everybody knows pea, and that is Pisum sativum. Pisum is the common name, and sativum is not common anymore. There are so many new varieties of pea that are being marketed in India. So basically all these plants have a particular kind of structure associated with their roots. All pulses belong to Leguminosae family. All pulses, urd, arhar, moong, cowpea, these are known by cowpea, chickpea, these are the English names, cowpea, chickpea, gram, etc. are legumes. Pea, which has the botanical name Pisum, genus name is Pisum. Okay, it is a legume. Then there is a plant. Whenever you examine the grass growing around you, you must have seen a small plant having this type of leaves. A small plant having this type of leaves. Clover. How many people have found this kind of plant growing among the grass? It has 3 leaves and maybe some red spots sometimes. Sometimes there might be some red spots like this. Have you seen this? Okay, some people are raising their hands, I think. You have seen this, yes or no? This is clover. Okay, this is a clover leaf. This is also a legume. So when you go outside and take this plant out from the grass, you will see that there are, when you take a look at the root, give me a second. All right. The brown color somewhat. All right, this will do. When you take a look at the root, you will see pink colored nodules on the roots that I just showed you now. So this clover is also an example of legumes. Okay, and clover is the most easy plant that you can find around yourself in your school garden or in your school playground, where you can see the leguminous roots. Okay, just wanted to show you this. All right, and these are pink colored root nodules. Pink colored root nodules. Okay, so let me see if I can insert a few images of this part here. For some reason I am not able to show you in a fast pace. That's okay. See this again, see the image again, and that should help you. Okay. So this is, when you wash it with water, you will see an image like this. When you wash it with water, you will see an image like this. Okay. So after that you will see the pink color. Some roots are very brightly colored, okay, and if they are not brightly colored, you might see that it's not easy to see that color. So when you don't wash it, it will look like this. If you don't wash it, it will look like this. But when you wash it properly, it will give this type of color. Now this red color or pink color is the same pigment that is present in our human blood, and that is called hemoglobin. It's the hemoglobin pigment that is present here, and that hemoglobin pigment, because it is being produced by the help of both bacteria and the plant, we call it by different name, and we call it leg hemoglobin. Okay. So these are the nodules. Yeah. Okay. You can also, yeah, one more thing. Everybody loves beans also. Beans also are included in legumes. Beans. Okay. And one more plant. Yeah. Everybody must love beans, right? So I guess this sparked your interest. Okay. I hope this sparked your interest. Okay. And one more example of this plant is kidney bean. Everybody loves rajma, I guess. Kidney bean. It's also a leguminous plant. Kidney bean. The botanical name of kidney bean is, I think it's, yeah, Phaseolus vulgaris. Rajma. Phaseolus vulgaris is the botanical name. Yeah. So that's also a leguminous plant. Okay. Are examples of legumes. Are examples of legumes, and they will show nodulated root and show nodulated root. Okay. Now let's write a bit about this nodulated root and all this. From the part the question is going to come. Okay. So please write now. These are symbiotic nitrogen fixation examples. If any plant among these is given, then it will show symbiotic nitrogen fixation. Now what about non-leguminous plants having nodule? Non-leguminous plants possessing root nodules. Okay. So one plant is ulnus. Ulnus is a non-leguminous plant, and it shows association with a non-rhizobium type bacteria, and that bacteria name is Frankia. Okay. Ulnus, a non-leguminous plant, a non-legume shows association with Frankia. Symbiotic bacteria, okay, a symbiotic bacterium. All right. So this is about Frankia. So Frankia is very common. Repeatedly in NEET examination, a question has been asked. Name a non-leguminous plants showing symbiotic association. So please remember this. Rhizobium, everybody remembers. Rhizobium has become a very easy question. Everybody knows that rhizobium shows association. So normally the question that will come from rhizobium will not be a direct question. But because rhizobium is so common, so now the favorite question of the examiner is to ask about non-leguminous plants showing nitrogen fixation. So Frankia is a bacterium which fixes nitrogen for ulnus. Okay, so I hope this settles it. All right. So this is a non-leguminous plants. Okay. All right. One more thing. Now, non-symbiotic nitrogen fixation. The last question from this part is non-symbiotic or free-living nitrogen fixation. Free-living nitrogen fixation. Okay. So there are some bacteria and they are classified under two categories, whether they are aerobic or whether they are anaerobic. This is a next part of that question that is coming in the examination these days. So one is free living nitrogen fixing aerobic bacteria. Under this part there are some free living nitrogen fixing aerobic bacteria like aerobic bacteria like Azotobacter and Bacteria. Azotobacter and Bacteria. Let me check the spelling if I have done. Yeah, Bacteria. Yeah, exactly. That's the spelling. Yeah, exactly. Okay. BIA. So these are two bacteria which has been asked. Azotobacter has been asked many times, so I can guess that the next question, if he changes this question, he might ask about Bacteria. So Bacteria might you can say that it is important for the upcoming NEET examination. Okay. Azotobacter has been already asked many times, so this means Bacteria will be asked next, probably in this examination. Okay. And then there is another bacteria which is free living anaerobic bacteria. Free living anaerobic bacteria fixing nitrogen. Okay. And this example is under, comes under, yeah, the name is rhodospirillum. Rhodospirillum. Okay. Now there is one very tricky question from this. It becomes slightly tricky if you don't pay attention. Free living anaerobic nitrogen fixing bacteria, right? Rhizobium is also a free living bacteria and it can fix nitrogen also, but in what condition? If you have seen the past year question, you will maybe try to understand what I am trying to say. Is rhizobium, just tell me yes or no, it's a bit tricky question that I am asking you. Is rhizobium a free living nitrogen fixing bacteria, or it is symbiotic nitrogen fixing bacteria, or is it both? I'll just write you the question so that you can remember, otherwise you might forget. It's an important tricky question. Rhizobium can fix nitrogen. Options are symbiotically only, symbiotically only, free living condition, in free living condition. Third option is both 1 and 2. Fourth, with non-legumes. What is the answer? Can you guess? What can be the answer to this? Take a good guess. Yeah, it's a tricky question. Take your guess. What can be the answer? Yeah, good. Very happy that you have done it correctly. Taming Long, you have answered correctly. Nitish, see, yeah, exactly. Rhizobium can fix nitrogen both in free living and symbiotic condition. Paul, okay, Paul, thank you. I will remember your name. Alright, exactly, good, very good. Itanagar and Tamilong Shillong students. Yeah, it's a bit tricky. Rhizobium can fix nitrogen symbiotically, but there is a condition when you talk about free living. If I make this question more challenging, it will be free living on top of soil, free living below soil. The answer will be, then the answer will change. Then it cannot fix nitrogen when it is above soil. On the soil surface, it cannot fix nitrogen. It can only fix nitrogen when it is below the soil, because the nitrogenase enzyme of rhizobium is poisoned by atmospheric nitrogen. That is why in free living condition, if it is receiving oxygen, it will not be able to fix nitrogen. Only in anaerobic free living condition it can fix nitrogen. There is a next level question, even I can make this even more difficult. I can change the option to free living anaerobic, free living aerobic. The answer will be symbiotic and free living anaerobic condition only, if I want to make this more difficult. So that is going to be the answer. Alright. So I'll explain now as I'm moving on, that rhizobium can also fix nitrogen in free living condition, but that will be in anaerobic condition only. Anaerobic condition. Also Frankia can also fix nitrogen anaerobically in free living condition. You can also write Frankia. Rhizobium and Frankia, in anaerobic condition, they can fix nitrogen. But in aerobic condition, they will not be able to fix it. Why? Because nitrogenase enzyme is poisoned by oxygen. Okay. So this part is done. I am happy that some people gave me the correct answer for which I desired. So this is good. That it means that you are making progress. It means that you are making some progress, and I'm happy that this is the case now. All right. So let me continue. Now I have to discuss two processes. First, I have to discuss how symbiotic nitrogen fixation in rhizobium is taking place in the bacteria. So first try to understand what is happening. First of all, the root here will come into contact with bacterium. Immediately as it comes into the contact with bacterium, it will curl around the bacterium. If the bacteria is present in the soil, so as soon as the root touches the bacteria, it will coil. Okay, it will show a curling movement, and in this curling movement, a lot of bacteria, they will get entangled, they will get attached, they will not flow away with the water or by anything else. So in this condition, in coiled root condition, the bacteria size is very small. Okay, it will start binary fission, and it will go on multiplying inside the root hair. If you remember the chapter of anatomy, after the root hair layer or the epidermal layer comes the cortex layer. So from root hair epidermis layer, it will now reach the cortical layer. Bacteria will continue to divide, it will reach the cortical layer, and as it reaches the cortical layer, now it can go into the inner part. When it reaches the pericycle, cortex, endodermis, pericycle. When it reaches the pericycle, now the root hair, what does it do? Now the bacteria, what does it do? It secretes some growth regulatory chemicals. These are basically hormones. Auxin and cytokinin, they are released, and they are synthesized with the help of the bacteria. Basically, bacteria manufactures auxin. Cytokinin is already manufactured by the root. I hope you know this, right? Do you know this? Yes or no? Auxin is manufactured by which part of the plant? Auxin is manufactured by which part of the plant? Yeah, I think everybody knows. That is the apical meristem. But cytokinin hormone is manufactured by the root. I'm not sure if you would have known this. And auxin and cytokinin, whenever they are present together, they cause rapid cell division in plants. That is how tissue culture is done. So plant is making cytokinin, bacteria is making auxin. This causes sudden growth explosion, growth of cells in a rapid manner, in a tumorous manner, and this forms a root nodule complex. From the pericycle and the cortex, new cells will start dividing, and they will form a round structure because the growth is indefinite, so it will occur in all directions. Cell division will occur in all directions. This is the basis of formation of root nodule. Now in this root nodule, the bacteria is going to enter inside this root nodule, and it will now sit inside the root nodule properly. It has been captured by the plant, or basically it has entered the plant by its own will and by the plant's will, and they have traveled to the cortex, they have traveled to the inside, the vascular strand, and now when it is sitting in the cortex, sitting in the pericycle, the bacteria establishes connection with the phloem. Why will it establish connection with the phloem? Because later on it will manufacture something for the plant, and in return the plant will give sugars to the bacteria, food to the bacteria, water to the bacteria. Nutrients will be supplied by bacteria forming vascular connection both with xylem and phloem. Now the bacteria will manufacture a part of hemoglobin, and the half part of hemoglobin will be manufactured by the plant. They will join together. This forms leg hemoglobin. Leg hemoglobin acts as oxygen scavenger. No oxygen will be able to enter this root nodule without being captured. So in the root nodule, the effective oxygen concentration will decrease rapidly, and now the bacteria which has the dormant enzyme nitrogenase, it will become active. It is no longer poisoned by oxygen. It will start working. It will take the atmospheric nitrogen that is present everywhere. Oxygen will diffuse into the nodule, and from this nodule, the nitrogen will be fixed in the form of ammonia. And there is a chemical reaction that is given in NCERT. Everybody knows it. Okay. So ammonia will be formed. From this ammonia, we will make ammonium ion. This ammonium ion will be given to carboxylic acid to make new amino acids, or it will be given to existing amino acids to make new amino acids. This is called transamination and amination process, and this is how nitrogen in air in inorganic form enters the plant in the form of organic substance. This is the whole story that I have told you. Okay, so now we can begin the process. Let's finish it quickly and let's finish this chapter now. Please write first step. The first step in nitrogen fixation in legumes is nodule formation. Nodule formation. Okay. Now this nodule formation. So let's discuss the nodule formation steps. What is the steps of nodule formation? Let's give it a subheading. Let's give it a subheading. Nodule formation begins by soil particles having rhizobium coming in contact with the root hair. Soil particle containing the rhizobium bacterium coming in contact with the root hair. Soil particles containing rhizobium. Okay, so let's denote the bacteria by red color. Rhizobium bacteria. There is another question in your exemplar, that what will happen if a leguminous plant grows in a soil having no rhizobium bacterium? What will happen if a leguminous plant is grown in a soil containing no rhizobium bacterium? It will not grow properly. It will show symptoms of nitrogen deficiency. Why? Because there is no rhizobium bacterium. So it will most probably show symptoms of nitrogen deficiency, because leguminous plants require very high level of nitrogen for growth. If it is not present in the soil, they will grow poorly and the growth will not occur properly. Okay. Now the root particle, they will basically curl towards the bacterium. They will curl towards the bacterium, capturing some bacteria along with it, and this bacteria gets entrapped in this hook-like structure. Bacteria, and now the bacteria will travel inside this hook-like structure. Like this. Infection thread containing bacteria. Okay, this is what will happen. Now moving on, you will say that the bacteria travel inside the root and reach the pericycle. It will reach the pericycle. This is the endodermis. This is the endodermis, and it will reach inside this part, pericycle part, and later on it will cause rapid cell division. Rapid cell division in pericycle. So this pericycle will give rise to something present on the surface, and it is pink in color. This is called the root nodule. This is called the root nodule. Okay. Now please write quickly. As soon as you have drawn the diagram, please let me know so that I can move further. As soon as you draw it, please let me know. Okay, yeah, sure. I'll scroll up. When you need me to scroll down, let me know. Okay, scrolling down. Done, I hope. Okay. All right. All right. So I hope this is done now. There is one more thing that you need to know. This question actually comes in NEET exam, but the thing is, they have deleted this information from your NCERT now. Okay. There is a small point that you need to know. There is a point about NIF gene. Whoever is solving the question must have come across this term, NIF gene. NIF gene is found in NIF gene cluster which is involved in, I'm giving this in a form of question because questions have been asked, which is involved in nitrogen fixation, are found in number one option is, are they found in? You haven't heard of it, okay. Yeah, it's because it's an old question. Previously it was in your this information present in your old NCERT or 3-4 years ago. Okay, so now they have deleted it, but it's okay. Question might come. Just try to learn this. It's a very short thing. Found in bacteria, symbiotic bacteria only. Second option is plant associated with bacteria. Third option is both A and B. Both A and B. D, in free living bacteria. Okay, so you haven't heard of it, so I'll tell you the answer. It is present in both bacteria and plant. Just try to remember it. It's an old question. NIF gene. NIF gene is responsible for nitrogen fixation, and it happens to occur in both plant and bacteria. Both have this gene cluster, and because of that only they have this close association. If this gene was not present in both of them, then nitrogen fixation will not happen. This also solves a particular mystery. Why do some plants fix nitrogen and others don't? Because the others which are not fixing nitrogen, they do not have this NIF gene cluster. However, if you insert this NIF gene into some plants, they will become nitrogen fixing symbiotic organisms. They will be able to form nodule. So this is being studied now. This information is being studied now. Just try to learn it. Okay. So NIF gene occur in both rhizobium and the leguminous plants. Okay, this settles it. All right. Moving on. Let me write a few things. The bacteria present in soil causes root hairs to curl, inwards as soon as they come in contact with it. As soon as they come in contact with it. Also, the bacteria divide and form an infection thread. The thread-like appearance form an infection thread. And reach the pericycle. On reaching pericycle, they secrete auxin. There they secrete auxin. And cause cells to divide rapidly, forming root nodule. Okay. Now next thing, the root nodule becomes pink in color because of leg hemoglobin. It becomes pink in color. Leg hemoglobin acts as oxygen scavenger. Leg hemoglobin acts as oxygen scavenger, okay, which decreases the concentration of oxygen in root nodule. It decreases the concentration of oxygen in root nodule. Okay. And because oxygen concentration has been decreased, now it can fix nitrogen. This is necessary because oxygen acts as poison for catalyst, acts as poison for nitrogenase enzyme. Because oxygen acts as poison for nitrogenase enzyme. Next point, molybdenum acts as cofactor for nitrogenase enzyme. Acts as cofactor for nitrogenase enzyme. Now the chemical reaction that happens inside root nodule nitrogen fixation is N2 plus 8 electrons plus 8 protons plus 16 ATP gives 2 ammonia molecules plus hydrogen. Hydrogen is a side product, it will escape quickly. 16 adenosine diphosphate plus 16 inorganic phosphate. Pi is inorganic phosphate. This might confuse you. This is inorganic phosphate because ATP is consumed, right? ATP has 3 phosphate. ADP has 2 phosphate. The extra phosphate comes out in form of PO4 minus 3. Okay. So this is the basic chemical reaction that you have to learn. So formation of one ammonia molecule requires how many ATP? 8 ATP. So this is another question in your NEET examination. Among the very easy questions, some questions are very easy in the NEET examination. This is one example of a very easy question. Formation of, if you need to scroll up, let me know, okay? Because I am writing, I think maybe a bit faster than you are copying. So if something is remaining, let me know. Formation of one ammonia molecule requires 8 ATP molecules. The name of enzyme involved here is nitrogenase. Okay, so this is done. Let's finish the last part. Fate of ammonia, fate of NH3. What happens to NH3? So there are two processes. It's already given in a book also. Fate of ammonia. Ammonia is used to make amino acids. I have a question for you. The number of essential amino acids for a plant is 10, 20, 2, 0. There are 4 options. So I want you to give me the possible correct answer. What is the number of essential amino acids required by the plant? In human beings, it is 10. Human beings require 10 essential amino acids. We can make all other amino acids from those 10 essential amino acids. So we can take only 10 amino acids, and every other amino acid will be made with the help of those amino acids. Now I am asking you this question: what is the number of essential amino acids for the plant? I have not finished fate of ammonia. This question is just an extra that I have just put here. Okay, Itanagar, you have answered. Tamenglong, you have answered. Shillong students, you want to take more time for answering this question? Okay, 2. Alright. Okay, most people are saying 2. Some are saying 20. Can you name those amino acids? Can you name those amino acids? The answer is none of these. Answer is 0. 0. The number of essential amino acids for any plant is 0. Why? Think about it. When you give a plant water, soil, are you also putting amino acids in the soil? Are you taking your blood out and performing some ritual? Plants are not vampires, right? They are not. I am not taking my blood out and giving to the plant that please take your essential amino acids through my blood. My blood contains a lot of amino acids. Are they eating you? Are they consuming something? Plants are not consumers. They do not require organic substances. They require only inorganic substances. Okay. So this was a trick question. Alright, I just wanted to test you. Yeah, I made this question. This has not come in the NEET examination. Not up till now. Maybe it can. Okay, so plants require only organic nutrients, inorganic nutrients, sunlight, water, all these things. Everything they will make by themselves. Okay, so I hope this is clear. Fate of ammonia. There are 2 main ways. Please write. There are 2 main ways. Yeah, all the essential elements are all inorganic compounds only in the soil. Okay. If that was not the case, we would have to give plants some blood of ourselves, okay, not just water. They would also require a blood. Okay, so that is not happening in any case. Okay, so even the plants, if they are insectivorous, they do not take organic constituents. They digest the insects. And convert them into inorganic substances, and those inorganic substances are absorbed. They never take organic compounds. Plants are never consumers. Okay, you should not consider them as consumers. However, I can give you some examples of other parts also, but they will absorb as inorganic compound only. So there are two main ways in which NH4 plus or ammonia is used to make amino acids. In plants. First is called reductive amination. Reductive amination. You can say here, in these processes, ammonia reacts with alpha ketogluteric acid and forms glutamic acid. As follows. It's a short reaction, just write it quickly and let's finish it. Alpha ketoglutaric acid, alpha KGA plus NH4 plus NADPH2 gives, in the presence of enzyme, glutamate dehydrogenase. It forms glutamate plus H2O plus NADP plus, alpha ketoglutaric acid plus ammonium NADPH2 reducing agent. This is the first thing. Second is transamination. Transamination means you take amino acid from one product, one reactant, transfer it to another, and the other thing that is receiving this amino acid is usually another carboxylic acid, and this carboxylic acid, after receiving amino group, becomes a new amino acid. So this is called transamination, basically transferring the NH2 group. Transamination. What is happening here? There is an amino donor, amino group donating compound R1C, NH2 group receiving H plus ion becomes NH3 plus H COO minus plus R2 C double bond O COO minus. This is amino donor, this is amino acceptor. Combine them together, and this is a reversible reaction, it can occur both ways. This will give, first compound will convert into carboxylic acid, so R1 C double bond O, keto acid actually, COO minus, plus second compound converts into amino acid, R2 CCOO minus NH3 plus H. This is a new amino acid. Okay, this is a new amino acid that is formed by transamination. This is how we make new amino acids, or in our body also transamination happens. We take essential amino acid, and essential amino acid combines with carboxylic acid and makes non-essential acid, non-essential amino acid. Okay, so non-essential amino acid is made by the help of essential amino acid. So you make one and you can make others out of it. That is the theory behind this. Transamination. Okay, so you can say what happens in transamination: transferring of amino group, transferring of amino group from one amino acid, from one amino acid to the keto group of keto acid, to the keto group of keto acid. Keto acid is converted into amino acid. Okay, so this is the part, last part of this chapter. Now there is another thing, nitrogen transportation. Nitrogen transportation, a small subtopic. How is nitrogen transported as? So you can write here, nitrogen is transported through xylem. Nitrogen is transported through xylem, mostly as amides. Mostly as amides. Exception, there is an exception to this. Exception. Soybean plant transports nitrogen as urites. Transports nitrogen as urites. Okay, So your chapter is finished. Mineral nutrition is now finished. Now are you ready for the next chapter? I hope you don't need a break now because the class is about to get over. About small time remaining. Yeah, everybody is exhausting right there on the table. Yeah, you don't want me to start the next chapter? Yeah, the next chapter is photosynthesis. Okay, take a break. Take a break. After this, yeah, today I'll give you these notes on your group as PDF, okay, because this chapter is finished. So when the chapter is finished, I can give you the notes, okay. So now I will give you these notes in the form of PDF, okay. Alright. So take a break. Okay, so let's begin the next chapter. Please write the heading photosynthesis. I should use green color actually when it comes to photosynthesis. So let's start photosynthesis. All right. So what do you mean by photosynthesis? What is the meaning of photosynthesis? It basically relates to synthesis of food materials in the presence of light. Photo means light, to make in the presence of light, to make food in presence of light. What do you make in presence of light? Food. So that is obvious. Now photosynthesis has a long history. We must actually discuss some part of the history of photosynthesis, of photosynthetic reaction. Everybody knows the chemical reaction of photosynthesis. 6 CO2 plus 12 H2O gives 6 H2O plus 6 oxygen plus 6 H2O. Sorry, plus C6H12O6. So everybody knows this chemical reaction. That is fine. But what I want you to understand is that photosynthesis chemical reaction was studied by chemists and not biologists. Okay, there are no biologists behind the study of photosynthesis. Calvin was a chemist. The man who gave the chemical formula, and the discovery of oxygen, that was by a chemist, and that same chemist performed some experiments here. So not much biologists are behind the discovery of chemical reactions of photosynthesis. So first of all, we will study some basic ideas how photosynthesis established as a biochemical redox reaction history. So there was some scientists who established that there is an experiment. I hope you know this experiment. It's called Priestley's experiment. Priestley's experiment. What did Priestley do? He took a bell jar, glass bell jar, and he placed a mouse inside it. He placed a mouse inside it, and what will happen to that mouse? It's anybody's guess. Okay, so the mouse, after some time, covered with a bell jar, the mouse died. The mouse actually died. Alright, so I'm trying to just copy him. Okay, so you're right. So the mouse became a bit bigger. That's not my intention. Actually, I'm trying to show something else. All right. I'm unable to show it. Okay. Anyway, the mouse basically died. Okay. The mouse died. And on that basis, you can say that the air inside the experiment is obvious. The air inside the bell jar is limited. Next, you repeat this experiment, and when you are repeating this experiment, you place a plant along with it. Repeat this experiment, Actually, the bell jar is not bigger. I'm just making some space out of it. Okay, and next you also place a plant next to it. Sorry, wait a second. I made an error. You repeat this experiment, but not with a plant. First of all, light a candle in here. Light a candle in here. And now both the candle will extinguish fast and the mouse will also die fast. Okay. The mouse will also die fast and the candle will also extinguish fast. Now you repeat this experiment, repeat this whole experiment, and now you place a plant next to it. A green potted plant is placed next to it. So now what will happen? Now what will happen is, place all this arrangement under sunlight. Place all this arrangement under sunlight. Like this. Now the mouse is going to live longer, the candle is going to burn longer, and this was Priestley's experiment. This was Priestley's experiment, that when he performed this arrangement, he saw that all of the things are living longer. Okay. So on this basis, he concluded that both both animals when breathing and substances while burning, they give out impure air, and plants take the impure air and convert it into the pure air. So basically, this is giving impure air, this is giving impure air, and in return, the plant is giving pure air to both. That is why they are surviving. Plants take. Priestley's experiment proved that Priestley's experiment proved that. Look, people at that time were not familiar with carbon dioxide and oxygen concept. Priestley, along with another scientist, Lavoisier, they basically what they did was they basically discovered oxygen. Priestley was the main man, and he was a chemist. Proved that plants give pure air, or they purify air and take in impure air. Dephlogisticated and phlogisticated air was a term used by him. Impure air. Animals, on the other hand, animals while breathing and substances while burning, release impure air, release the impure air and take in pure air and take inside pure air. So this was Priestley's experiment. Okay. After Priestley, there was an experiment by Saussure, Nicolas de Saussure, proved that amount of impure air taken by plant, impure air taken in by the plant is equal to amount of oxygen released by the plant. Okay, so this is the thing. After this, there were some other experiments, and you know about these experiments very, very well. I hope you've written. Can I scroll down now? What were these experiments? These other experiments include taking a plant and put it in dark, okay, and after 2 days taking that plant again and inserting one leaf of that plant in a bottle, and in that bottle potassium hydroxide has been placed. Potassium hydroxide will absorb all the carbon dioxide in that bottle, and now if you put this plant in sunlight, what will happen is this plant will not be able to make starch. Okay, carbohydrates, there are many experiments. Experiments which prove carbon dioxide is essential for photosynthesis. Experiments which prove chlorophyll is essential for photosynthesis. For this experiment, you need a potted plant which has some yellow spots or any spot other than green color. Yellow spot, white spot, or red spot. This is called variegated leaf. Variegated leaf. If you perform a starch test, you will see that starch is being made only in green portions of the plant, not in green portion, not in non-green portion. Only in the green portion of the plant starch will be manufactured. Iodine leaf experiment. Okay, iodine starch test. Also, oxygen is made. The same Priestley's experiment. This same Priestley's experiment is being repeated in such a way. What is happening is oxygen is being made, and how do you prove that it is oxygen? You take a cindering, like you take a mastic and it's just about to be extinguished, and you take this glowing mastic in the jar full of oxygen. So it will suddenly burst into flames. So there are some other experiments which prove oxygen is made in photosynthesis, carbon dioxide is used. On these informational basis, you derive that the equation of photosynthesis looks like this. That the raw materials for photosynthesis are carbon dioxide. Okay. Please note that I have not put water in here. I am just saying raw material for photosynthesis is carbon dioxide, and chlorophyll and sunlight are essential. Chlorophyll and sunlight are essential. Also, we know that plants use water, but I am not putting water in the equation. For one reason. I will just show you. And starch or sugar is made in photosynthesis? Starch plus oxygen is made in photosynthesis. This is finished product or final product of photosynthesis. So if you try to write the equation, it will become like carbon dioxide plus I don't know what gives C6H12O6. Glucose is the monomer of starch. First glucose is made, then starch is made. So I'll just write C6H12O6 plus oxygen. Now you see in the left side of the equation there is no hydrogen. There is no hydrogen. So we need a hydrogen donor substance. So there is only one common hydrogen donating substance, and that is called water. Water can be the only possible hydrogen donor. So if you now try to balance this equation, it will become 6 carbon dioxide plus 6 H2O gives 6 oxygen plus C6H12O6. This was the first equation. Okay, this was the basic idea of the equation. Later on, this equation was modified to give 12H2O C6H12O6 plus 6O2O. Reason is, I'll show you the reason. There is a scientist called Van Niel. Van Niel studied sulfur bacteria and found that they use hydrogen sulfide gas instead of H2O and make carbohydrates. Okay, so what did he do? He found that in sulfur bacteria the equation looks like this: 6 CO2 plus 12 hydrogen sulfide gives C6H12O6 plus 6 H2O plus 12 sulfur. Now if you compare this equation with this equation, compare this equation with this equation, what is missing? Van Niel said that if hydrogen sulfide is giving sulfur, so this means hydrogen donor is being broken down. Hydrogen donor is being broken down. So after that the equation, therefore equation of photosynthesis becomes, I'm just finishing it, just give me 1 minute, okay, becomes 6 CO2 plus 12 H2O gives C6H12O6 plus 6 H2O plus 6 oxygen. So it was basically estimated that the equation of photosynthesis should be like this, based on information provided by Van Niel. In the next class, we will study how this was confirmed that oxygen comes from breaking down of water only. Rubin and Kamen's experiment, okay, and Engelmann's experiment. We have to do a lot of part. So the next week, in the next chapter, we will discuss light reaction, dark reaction, and all these things. Okay. So that's enough for today. Okay. So thank you, dear students. Thank you for your patience, and enjoy your time. Enjoy your weekend. Study a lot. I'll give you questions, and the notes of this chapter I'll give you in PDF format on your WhatsApp groups. Okay. I'll do that today in the evening. Thank you. Have a great day. Thank you.